I've been interested in infectious diseases for a very long time. When I was at the Institute for Genomic Research back in the late 90s and early 2000s, Olga mentioned that the first genome was sequenced there. Technically speaking, the first bacterial genome ever sequenced was sequenced at Tiger, published in 1995 by Craig Venter and Ham Smith. And I wasn't part of that. I joined 2 years later. But that's where a lot of the genomics revolution got started. The Human Genome Project was well underway then. But what I want to talk about today is my vision of how I think next generation sequencing can revolutionize the treatment of infections. So let me give you a little background. So sequencing today, as you all know, I think, is vastly more efficient than it was in 2001. I think 2001 is a landmark because, as you'll see on the next slide, that's when the human genome was published. Many more than half of infections in the US go undiagnosed. I'll come back to this a little bit later. But as you all know, I'm sure all of you have infections. No human being can live beyond a few years without getting an infection of some type or another. Vast majority of infections are not ever positively diagnosed. If you have kids, take them to the doctor, they have a scratchy throat or a runny nose. The doctor might guess that they have a viral infection or might guess that they have a bacterial infection. The vast majority of times they will not have any positive diagnosis. If they think it's bacterial, they'll give you a broad spectrum antibiotic and send you home. And that's frequently incorrect. If it's viral, of course, that doesn't do any good. But we have this tremendous tool at our disposal now, which is DNA sequencing. And I think we should be using that even in those trivial cases. I'll talk a little bit later about even in more critically ill patients in the hospital with infections, we're still doing a very poor job in more than half the cases not positively diagnosing their infections either. So we have the technology to do something about this, and I want to explain to you how. And we've started to do it in an experimental way at Hopkins. So let me go back a little bit to look at the technology and where we've come. So this was, you've probably all seen these covers before. On the left is the issue of Science that the Human Genome was published in 2001. On the right is the issue of Nature. They were published simultaneously in the middle of February. And there were, for those of you who were following at the time, there was a very exciting race to finish the genome. The Science paper was done by a private company called Celera Genomics. And I was collaborating with them at the time. I wasn't at Celera, but it was a spin-off from Tiger where I was. So that included everybody at Celera and several dozen outside scientists. And then the Human Genome was the culmination of the effort started in 1989 to sequence the genome by a large international consortium. So the Human Genome was published. The technology used at the time was this machine here, the ABI 3730. This machine was announced the first version was announced in 1998. And that's what really led to this race beginning, because it was announced by the company that funded Celera Genomics. And it was a breakthrough at the time. It was the first time we were able to sequence DNA through capillaries instead of in a big slab gel. And it allowed you to get much greater throughput at lower cost than had ever been possible before. And it was advertised as being able to do up to 1 million bases a day. Actually, that's a later model. This is the first model wasn't even that efficient. And we thought that was great. I was part of the planning team that when we were figuring out what to do with these machines. Even though I didn't join Celera Genomics, I was part of this team that put the plan together. And that's what made a number of the scientists, most notably Craig Venter, realize, oh, you could sequence the whole human genome as a shotgun sequencing project with this kind of machine if you could put enough of them together. And that was considered radical. And the read lengths got to be up to over 800 base pairs long. And the costs at the beginning, they were a little more, but by 2001, it was about $1 per read. So a read is 800 base pairs of DNA. And that was considered amazing. That was 15 years ago. So today, the state of the art is the Illumina HiSeq 4000, which can go and generate sequences at a rate of 400 billion base pairs a day. And it costs about $1 to generate 62 million bases. So 2001, we had 1 million bases a day. Today, we have 400 billion. This is not a small increase. So it's 400,000 times faster. As far as I can tell, looking at other technologies, we've never had any technology, any man-made technology, that's improved at this rate over this short of a time. We used to think, I was originally trained in computer science, we used to always point to computer processing speed as look how it's getting faster and cheaper every year, and it's amazing, look how transformative it is, and that's true. But sequencing technology like this is also transformative, and it's happened so fast that many people, even people working in genomics, haven't realized what it allows you to do. So we're constantly trying to think of, well, now what can we do that we couldn't do just a few years ago? It also got a lot cheaper. So $1 for 800 base pairs versus $1 for 62 million base pairs, that's 77,000 times cheaper. So I tell my colleagues frequently that they should think of sequencing as free, because it's not free. Okay, it still costs money, but compared to what they were thinking of if they were doing sequencing 10 years ago, as many of them were, it's free today. Just don't think about that as a barrier. Everything else you do is going to cost more than the sequencing part. So think about what you could do if you can get as much sequence as you want for nothing. And then if you really want too much sequencing, maybe it won't seem free. So one other way to put this in context, if I wanted to sequence a human genome in 2001, I would need to put together 8 of these sequencers and run them for 8 months at a cost of $30 million, and I would have enough to assemble one human genome.
Is that 8 sequences or 100?
That's 100 sequencers.
Yeah, I thought you said 8. Okay.
Sorry, 8 months. 8 months. That's how long, if you ran them all flat out for 8 months. Today, you can run, with that machine, you can generate a genome in a day for about $1,500, which is not free, but compared to $30 million, it seems free. And in fact, the real costs are lower than that. That's what you'd have to pay if you own the machines. But anyway, it has gotten incredibly cheap to do sequencing. So, all right, so how can we use this to transform medicine? The human genome, when it was published, by the way, one of the many things it was claimed was it was going to transform medicine. And that's happening, but it's been 15 years and it hasn't happened maybe quite as much as people were expecting. I'm not going to talk about human genetics today. I'm going to talk about bacteria and viruses, though, instead, and how we can transform that. So there's another thing that has happened that we have used these sequencers for over the past 15 or 20 years, and that's we've done a lot of pathogen genome sequencing. So as I mentioned a few minutes ago, the first bacterial genome was published in 1995, which is really very recently, only 20 years ago. And that was a big deal. That was Haemophilus influenza. It was 1.8 million bases long. People thought, wow, that's amazing. You could do a whole bacterial genome. And right away, people started doing more genomes. In fact, the second and third bacterial genomes that were done were also done at Tiger the following year. So a year later, we had 2 more. And by the end of the 90s, we had about a dozen, or maybe 20. And then things really got going when we got to next-gen sequencing. So today, basically all known human pathogens have been sequenced many times over. We have many strains for most bacteria and viruses. There are over 40,000 bacterial genomes that you can go and download today. Not all finished genomes, so many of these are draft genomes, which means that they're in multiple pieces and there might be gaps in the assembly. There are about 4,000 that are finished, meaning the entire sequence has been filled in. And there's over 80,000 virus genomes, and actually these are underestimates, but these are easily available publicly. And there are hundreds of eukaryotic pathogens. Eukaryotic pathogens are things like the malaria parasite, and they're a lot bigger. Some of them are much bigger than bacteria, but we have hundreds of those as well. So those have all been sequenced, so we have used the power of sequencing to create all this data. And I've been involved in a lot of these projects, not thousands, but I've been involved in many bacterial sequencing projects, many viral sequencing projects. And I can tell you from the point of view of someone who's doing the sequencing, we were all doing it thinking, well, once we've done this, cures and treatments for these diseases, these organisms, the diseases these organisms cause are going to come soon. But they haven't, all right? Not in many cases. But one thing we can do, I think, is we can use them for diagnosis. And I think we can do that today. So I want to try to convince you that we can and show you some examples of how we've done it. So what I'm here to tell you about is how we can use sequencing to do diagnosis, direct diagnosis of infections from sequencing. So the idea is very simple, really. Once you consider sequencing to be cheap or free, and once you realize that we've already sequenced all the known pathogens, if I can get a sample of infected tissue from a person, then I want to just send that tissue for rapid sequencing using a HiSeq or a MiSeq, 2 of the common instruments these days, generate tens of millions of reads. A MiSeq run will give you 20 or 30 million reads. And that's going up again this year. And then look at that data and search that data to find the infectious agent. Okay, so if I just take a sample from a person, and I'm going to show you some detailed examples. If I take a sample from a person and sequence it, it's going to be mostly human DNA. And when I tell this to colleagues, especially clinicians, they're like, well, that's just going to be human, and there might be a few bacterial cells in there, but isn't the human going to dominate the sample? Yes, the human sequences will completely dominate the sample. They'll be 99.5 or 99.9% of it. But that doesn't matter, because I'm going to throw that all away. Because remember, sequencing is free. So if I have 1/10 or 1/100th of 1% of the sample that represents the pathogen, that's plenty to identify the pathogen with high confidence. it's just disturbing to some people to think that, well, you're going to have all that human DNA that you don't use. And my response is, yes, we are. But it doesn't cost very much to do this, and we can figure out what the person's infected with. So we're going to generate a big data set, and then we're going to try to figure out what someone is infected with from that. So here, now I can talk about what is sort of nearest and dearest to my heart, which is computation. So we have to do that analysis very quickly, so let me talk about that for a few minutes. So if I'm going to generate, let's say, 10 to 50 million reads, what I'm going to do is I'm going to take each read. So these reads are going to be very short with next-gen sequencing. They're going to be 200 or 300 bases long. Some of them are going to be 100 bases long. And I need to take each read and compare it to all the thousands of genomes that have been sequenced. And I need to do that very quickly because there's millions of these reads, so it has to happen quickly. I want an answer quickly. So the standard way to do alignment for the past 25 years actually is a program called BLAST, that I'm sure many of you have used many times. It's very reliable. It's very sensitive. It gives you a detailed alignment showing you how a sequence matches a database of sequences. And there's a version of it called MegaBLAST that's pretty fast. And that can do this task for a large database of pathogens at about 7,000 reads per minute, which is really fast. If you were doing one alignment, by the time you hit the carriage return on your keyboard, you'd have your answer. You wouldn't notice. It would be milliseconds. That's not fast enough for this much data. So for this much data, even at that speed, it would take 3 to 5 days to align one sample. And of course, you could say, well, we can run computers in parallel, so we'll just throw 100 processors at it, and then it will only take 1/100th of that. That won't be too long, so maybe a few hours. But that's not good enough for me. So I want to be able to do it on one computer and still get an answer quickly. So can we get an answer to this question in 15 minutes? We have one read. We want to align it to, we have 10 to 50 million reads. We want to align them all, and we want to know the results in 15 minutes.
How much error? Depends how much error is how much noise, right?
We don't want much error.
So no, but if the design sequences, why it takes so long? It's very simple algorithm. Why so long?
Well, I'll show you some statistics. The accuracy is very high. But, well, if I'm trying to diagnose an infection, may not have very many reads from that infection. I'll show you some concrete numbers a little later, and you'll see that you don't want to miss the true positives, but you don't want to have many false positives either. So you need to be both precise and sensitive. So let me just talk about how the algorithm is very simple. So how to align species from raw reads, how to identify species from raw reads. So the usual method I just said is to use BLAST, which will align each read to a database. But I think that's too slow. So our strategy is much simpler because we actually don't need an alignment. We don't need to know exactly where the read comes from on the genome. We just need to know that it matches it. So we can insist on an exact match of some length k, and then look for that. And that's much faster. So that's our strategy, and I'll show you in like 2 slides how to do that, how to do it quickly. So the idea is we're going to take we're going to build a database of all the sequences of length k, or k-mers, in a set of genomes in a database. And we're going to record where those k-mers came from. And then for each read, we're going to look at each k-mer in the read and figure out 31.
31.
Yeah, but you can adjust that. Okay, so k needs to be long enough that you don't get accidental matches. So there's 4 to the 31 possible k-mers of DNA, so that's a big space.
Sequencing without error. Actually, sequences can really impact how much error.
Illumina sequences have an error rate of about 0.25% or lower, so they're very accurate. They do have errors, though. So we use this strategy in a program called Kraken that we published a couple of years ago. I'm going to give you the 2-minute or maybe 5-minute description of how it works. So the idea is we're going to identify, we're going to classify reads, label them with a species or taxon ID using short exact matches. That's a picture of a Kraken over there, but that's not our program. It's much more colorful than our program. But the program's called Kraken. So we're going to build a database for this problem. We've built a database with on the order of 4,000 bacteria and 40,000 viruses. We keep changing this database, but you can add whatever you want to it. And because of the task we're doing, we're going to also include the human genome in that database, because we want to take the read. Most of the reads are going to come from humans, so of course we need to have all the k-mers in human in our database. So just for those who aren't computational types in the audience, just what I mean by k-mer is suppose that my read is across the top, A's, C's, G's, and T's. And I suppose I'd choose k equals 10. That's not what I use for the program. We use 31, but you can pick your number. Then the first 10 bases of that read would be a k-mer of length 10. And I would shift over by 1. The next 10 bases, starting at position 2, that would be another k-mer. So from this read, I'm going to extract all the k-mers of whatever length k I've chosen. I know I can hear my microphone going in and out, so I'm trying to speak loudly so that when it goes out, you can still hear me. Okay, so the first step is to build, and you do this offline. So before we do any classification, we build a k-mer database. Again, pretty straightforward, but the idea is we pick some length k, and we go through every genome that we've decided to include in our database, and we pick the first 31 bases out of E. coli, and we have to store with that 31 bases the identifier E. coli. So we already, the microbial community has already handled this. Every organism that's been sequenced has a unique taxonomy ID or taxon ID. So that identifier, which is just a number, is going to be associated with that k-mer. In fact, for E. coli, there's hundreds of strains known. Each of those strains has its own taxon ID. So I'm going to take my k-mer and I'm going to associate with it. I'm basically making a big table, a hash table. And I'm going to have in the table the k-mers and the taxon IDs that they came from. I can hear this going out again. Might be me. So some k-mers are not unique to a species. And what I've tried to illustrate here is if you have a k-mer, and I've shown one so here's one in red that came from Salmonella enterica. So if that k-mer is only in Salmonella enterica, then I'll store the identifier for Salmonella enterica with that k-mer. I have an orange K here to indicate in a cartoon fashion the k-mer that came from both, that appears in both Yersinia pestis and Salmonella enterica. I only want to store one identifier with each k-mer to make things efficient. So what we do is we just pick the lowest common ancestor of those two from our tree of life, and that's Enterobacteriaceae. And there's an ID for every internal node in this tree, so I store that taxon ID with that k-mer. And I might have a k-mer, I don't know if any such k-mer exists, but I might have a k-mer that's common between Salmonella and H. pylori, which is much more distantly related. Then I'd have to go higher up the tree to Proteobacteria, and that's the taxonomy ID that I'd use. So that's the database construction process, and we then store them in a very fast hash array that uses another program called Jellyfish that I'm not going to talk about, but we basically build this big hash array that we can search very, very quickly. And for any k-mer, we can look it up very quickly in constant time and find out, well, what is the taxonomy ID that k-mer came from. And the majority of, if you pick K long enough, the majority of them will only come from one species. When you're down at the strain level, I haven't shown that here, but many of these common bacteria have multiple strains, and those are very similar to each other. So a lot of the k-mers are not unique to any strain, but will be at the species level. So that I do offline. And then when I'm doing classification, when I've got my data set, I've already done this part, I classify the reads. So here's my one-slide explanation of how the classification algorithm works. So I have a read, which is shown graphically at the top here. And I'm going to just go through that read, and I'm going to pick the k-mers out of that read, and I'm just going to look them up in the hash table. So it's very fast, constant time. So what I've shown here is some of them won't match anything. That's fine. As long as I get a few that match, I'm going to be OK. In fact, the way the program works is if we get even one k-mer that matches something we know, we'll spit that out as the identity of that read. So it's important that you pick K long enough that you don't get many false positives by doing that. So here I've shown with color coding where these k-mers match in this taxonomy tree. So the blue rectangles, suppose they all match this blue node in the tree, the orange K. So to figure out what this read is, then I will conceptually build a little tree of just the taxonomy IDs that have some hits. And I'll look at the number of k-mers that are found on each path from the root of that little tree to the leaf of that tree, and I'll find the path that has the biggest weight, and that's the path I'll use, and I'll report the leaf node from that path as the classification of the read. So that handles pretty much all cases. Most cases, most reads that we look at, all the k-mers, if they match at all, they all match the same thing. But once in a while, you do get these ambiguous reads, and if you get a read that's ambiguous, then it's going to be labeled with something higher up in the taxonomy. So it's not going to be useful for diagnosis. So we need to have some piece of a genome that's fairly distinctive or unique to that genome if we're going to do this well. But it turns out that most of the organisms that infect us are quite distinct from each other, and they're all very distinct from humans. So this is not a problem. So as it turns out, Kraken is very accurate. So here's we have many comparisons. I'm just going to show you one, showing you Kraken compared to a number of other programs, including the BLAST program, which is this more elaborate aligner. And what I'm showing here is on one particular complex data set, but it was an artificial data set, so we knew the true labels for every read, so we can give you precision and sensitivity. So sensitivity is around 80%, which is how many of the reads get labeled. And precision is in some ways more important. It's if you give me a label, what's the percentage of those that are correct? And that's near 100% for Kraken, and it's near 100% for BLAST as well. So we could use BLAST. BLAST would work just as well, but BLAST is very slow. So here's the speed. So Kraken runs this is on a single machine, fairly conventional machine. It runs at about 1.5 million reads per minute. So in fact, I can do 10 or 20 million reads in 15 minutes, whereas MegaBlast, as I said before, runs at about 7,000 reads per minute. And you might see there's a program called Metaflan here. It's not actually a classifier. Metaflan will not classify most of the reads, but it does. It's a program for trying to give you a picture of the composition of a population, and that's a little slightly different topic. There was a paper published earlier this year that I just thought I'd mention that compared. You might think we're not the only people who have thought of this task, so there's about a dozen different programs that do this. There was a paper that appeared this year that compared them all, and this is showing you the accuracy or the error rates of these programs. And I had nothing to do with this paper, so so therefore I like it, because Kraken had the lowest error rate and was one of the fastest tools. So the lower is better here. Okay, so we have a tool that can do this, but there are some complications. So with real data, there's a lot of things that come up that make that before you start just using this to figure out what a patient has, what a patient's infected with, there's some problems you have to be aware of. So one, these problems will create false positives or false and possibly false diagnoses, and you don't want to do that, certainly not with real patients involved. So one problem that happens a lot is we have low complexity sequences. So these are real. In genomes, we can have sequences that are, say, runs of all A's or CT, 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 like I've shown here. And that can really occur in a genome. There are some genomes that have these low complexity regions. And when a sequencer is just not getting much of a signal, it can also spit out low complexity sequences. So by chance, you'll get a match which isn't real. So we don't want that to happen. Another problem is that most of the genomes we're looking at are draft genomes, so they can contain contaminants. Those contaminants can and often do include human DNA, because it's humans who are doing the sequencing. And if they haven't carefully screened their data before they deposit it in public archives, there can be some human DNA that's labeled as something else. And that means that I'm going to get a match to that something else and think that something else is in my patient, but it's not. It's human DNA, which I already knew was in my patient. So we don't want that. So those are computational problems. There's some biological problems too, which is that I'm proposing to collect biopsies or samples of some type from people. There are bacteria and viruses all over us. They're on our skin everywhere. They're inside of us in various places. We don't know what they all are, but we've sequenced a lot of them. And we know they're there. And you might think that you could, well, if you're taking it from somewhere in the body, just wash the skin really thoroughly. That definitely helps, but it does not remove all the bacteria and viruses. Many of them live under your skin. So pretty much anywhere you go to collect a biopsy from a person, you're going to be passing through some bacteria and viruses. So those are going to be in your sample. So you have to learn how to recognize those and not incorrectly call them as a causal for an infection. And then finally, the sequencing lab itself is also not a sterile environment. There's bacteria floating all around. There are bacteria in their reagents, even ones that are supposed to be sterile. And that causes a problem. So I'm going to give you a little bit more detail about each of those for a few minutes. So, this is just to show you the low complexity data. This is real data from some of many false positive matches we got as we were developing this protocol. So here's some reads we got from a patient sample. The patient did not have Mycobacterium tuberculosis, but these were the matches. And when you look at them, you see, oh, those reads are just all Cs. So those are the reads that came off the sequencer, and they match. There's some place in that genome that has sequences that match this. There's basically a long run of Cs. So those are bogus. We don't believe those. And here's a similar kind of thing happening with Treponema pallidum. Again, the patient did not have Treponema pallidum, which, by the way, causes syphilis. But these were the actual reads that came off the sequencer, and they match because there are some regions of this genome that have similar low complexity sequences in them. So we have to screen those out. And there is at least one program called DUST that does a pretty good job at screening low complexity sequences, but it doesn't get them all. In fact, we ran that on all of our genomes before building the database, but some of these somehow slipped through. So that's one of the causes of false positives we have to worry about. I mentioned bacteria in reagents. So there was a very nice study done about a year and a half ago that looked at this question, because we all sort of suspect that there's bacteria in some of the reagents used for sequencing. So this group in the UK did this nice experiment. What's shown across the top are they did serial dilutions of a particular strain of Salmonella, an uncommon strain of Salmonella. So they pretended they were sequencing the Salmonella. They did sequence it. And then they diluted it by a factor of 10, and then another factor of 10, and so on until they had 10 to the 5th dilutions. And they sequenced it each time. And what they found was in the initial sequencing, when it was just Salmonella, that the vast majority of the reads were Salmonella. And as they diluted it, more and more, they still got the same number of reads, but the reads now showed this interesting mixture of bacteria. So these are heat maps showing you the different, these are, I think, family level. So the colors just represent different groups of bacteria. And the 4 different panels are 4 different reagent kits. So each kit has its own particular composition of bacteria that were apparently in the kits. And even when they were down with basically no salmonella reads at all, they still got plenty of reads from these bacteria just from the reagents. So this is a particular problem for what I'm proposing to do, because I'm looking at a problem where I'm thinking the vast majority of reads will be human, but I'm looking for a very small number of reads from the total sample. So even if there's only a few of them, I might think that's an infection. So the reagents are another problem. And you might think, well, you know, isn't that a solved problem to make the reagents sterile? And yeah, it is. You can just heat them up, and that will kill all the bacteria. But then if I ask, well, how do you know there's no bacterial DNA in the solution? I get blank stares from people who supposedly should know better. So we don't actually know how to get rid of all the DNA. We can kill the bacteria and kill the viruses, but their DNA is still floating around in there. So you can't get rid of all that. We don't have any little fine mesh net that will pick it out. So we don't have a good way of getting rid of all the DNA. Another interesting source of contamination is that some genomes contain bits of the wrong species. This was very surprising when we started to hit it, but we've hit it in a number of cases. We've cleaned up some of them. So a genome deposited as one species is usually that species. Actually, some of them aren't, but it's usually that species. But sometimes it has bits and pieces of others. So one that was particularly interesting from a couple years ago was this one. We wrote a little paper on it because we thought it was interesting enough to write up. And we got it fixed at GenMex. You won't find this genome is not in GenMex anymore, but we found that there was a strain of Neisseria gonorrhea that came from the Taiwanese CDC. That's what the TCDC stands for in its strain identifier. That had multiple sequences, 5 actually, that were from the cow and sheep genomes. 4 from cow and 1 from sheep. Each of them about 500 bases long. And we have no idea how they got in there. But it was not contamination in the lab. It was computational contamination. So somehow this group, in the process of depositing it in GenBank, had stuck in some sequences from completely unrelated organisms in it. And we actually found this because there was someone who wrote to me who thought that they were using the Kraken program in a cow metagenome project, and they found Neisseria gonorrhea. And this is actually a human-only pathogen, does not infect cows. And they said, there's no way this could be in our cow. What's Kraken doing wrong? And I looked and I realized that Kraken was doing nothing wrong, that in fact this genome was wrong. And there are others like this too. We found quite a few. And when we find one, it's usually pretty obvious, but it takes some digging to be sure. So now I've just told you why this is a problem. So I started by saying, well, we should be able to use sequencing and use these big databases of genomes to do diagnosis. So then I told you, well, here's all these problems we're going to have if we try that. So despite all this, can we diagnose infections with sequencing? So obviously, I think we can, or I wouldn't be here talking to you about this topic. I'm going to tell you next about some results from a paper we just published this summer in a neurology journal where we looked at brain infections. So the reason I worked on brain infections turned out to be a very good case. They're, first of all, very serious, and we really need to diagnose them. Patients will die quickly if they have a brain infection, and you don't do it, and you can't do anything about it. But surprisingly, they're not diagnosed that accurately. But another good thing about them, or a good test case for us, is that the brain is supposed to be a sterile environment. So unlike other parts of the body, say the skin, where you also get infections, there shouldn't be bacteria, we think, there shouldn't be bacteria or viruses in your brain on a normal, healthy brain. There might be, by the way. No one's proven that there aren't, but people sort of assume they're not. So we want to use next-gen sequencing, and we're now, for about 2 years now, been trying to do this at Hopkins, purely in an experimental, prospective way, to see how we can use sequencing to change the way we treat brain infections. So before I said 50% to refer to all infections, that was a gross underestimate. But for brain infections, it's closer to the right number. But surprisingly, even in the US and Europe, more than half of brain infections today, state-of-the-art, with state-of-the-art techniques, go undiagnosed, meaning they don't know exactly what the patient has. They often don't even know if it's a virus or a bacterium or something else. And one big problem is that, one reason this is so urgent is that viruses, bacteria, and autoimmune conditions can all cause very similar symptoms if they're in the brain. And proper treatment depends critically on knowing the right cause, even knowing which of those 3 it is. So for example, for an autoimmune reaction, my neurology colleagues say, well, you give steroids to suppress the immune response. That's the worst thing to do if someone has an infection, of course. So you need to know, is there an infection or not, before you do that. You'd like to know. But in more than half the cases, we don't know. So we built a database, a Kraken database, using this Kraken program. This was most of this work was finished a year ago. The paper just came out, but there's always some delays. We're still doing the work, but the database we used for this had about 2,800 bacterial genomes, about 4,400 viral genomes. We've now dramatically expanded that, so now we're up to 40,000, which are mostly strains of the same species. There are not that many new species, and only about 25 eukaryotic pathogens, and the human genome, of course. So I'm going to tell you about just 2 case studies for the next 10 minutes or so. And then I'll talk about a different application. So the first one, and probably our most interesting one, was a patient that we labeled PT8, because this is the 8th case that we looked at. So we got these at a rate of, about 1 a month, sometimes 2 a month, from our neurology clinic. So this is a 67-year-old woman who came into the clinic with a 2-month history of back pain, fevers, and dyspnea on exertion. And on diagnosis, they saw multiple intracranial and spinal cord lesions. I'll show you a picture on the next slide on MRI, and also lesions in her lungs. They did standard pathology testing, looking for bacteria, fungi, and looking separately for mycobacteria. That would be tuberculosis. And those were all negative, but they had one assay that was positive for a bacterium called nocardia. And based on that, they put her on several antibiotics, including ones that would treat nocardia. And she seemed to be getting better. She was discharged and went home, but then started getting worse again. They changed her antibiotic regimen when she was discharged, took her off all but one of the antibiotics, and she started getting quite a bit worse and was back in the hospital a few weeks later, when another brain MRI showed that the lesions were getting worse. So here's 2 different brain MRIs taken at the same time with just slightly different technologies. And what you see are, and here, hopefully there's no neurologist in the audience. Hopefully I won't say anything wrong anyway. But the white patches are not good. You don't want to see these. So these sort of white regions on the brain image are not supposed to be there. There's some kind of inflammation going on there. So you don't want to see, so it should all be sort of dark gray in the middle. The yellow circle shows you one of the many nodules. So right there in that yellow circle, that little white spot is one of the infectious lesions, and that's the one we biopsied, that very one right there. So a surgeon went in because they decided they wanted to do a biopsy. So in very serious cases, they do biopsies to get some of the matter. Sometimes these things turn out to be tumors. This clearly wasn't going to be, but they did not, I should emphasize, they did not do a biopsy for our sequencing experiment. They were going to do a biopsy anyway for pathology to get a better sample of what this patient might have had. And we, as part of our study, we took part of that biopsy material and we sent it for sequencing. So it turns out for this patient, we got 2 biopsies, one from right in the center of that little white patch, that's the nodular biopsy, S2, and one from the perimeter, that's the perilesional biopsy, or S1. And we didn't get a very good yield. Usually we get about 20 or 30 million reads. We only got about 7 or 8 million reads, or 6 to 8 million reads. So we did 2 different runs. We had enough material to do 2 runs on each of the 2 samples. So on S1, we got a total of 15 million reads, and we didn't see anything. After doing lots of filtering, we decided we could not see any signs of any infection we could name. But on S2, the sample that came right from the center of the lesion, we had about 14 million reads, and I'll show you the data. This is what we got. So this is after extensive cleaning, so I'm not showing you everything that we saw, but because we saw many, many things. But what we see is from the first sample, as we saw with all our samples, 99.7% of the reads, in this case 6.3 million, were human. The second sample, 99.7% of the reads were human, and that was 7.6 million reads. But then we saw 4 reads, just 4 reads from M. tuberculosis, 2 from P. acnes, 1 from Pseudomonas putida. Without going into lots more data, we see P. acnes in every sample we see. That is a really common skin bacteria. You just can't get it out of the samples, so we ignore those. We actually also see Pseudomonas in almost every sample. It's coming from somewhere. It's a pretty pervasive contaminant, but we did not see, this is our 8th actual case. We had never seen Mycobacterium tuberculosis before. These were 250 base pair reads. When you looked at them, you could see, when you actually did a more careful alignment, which we did with BLAST, you could see they're very clearly M. tuberculosis, and they don't match anything else. So there's no doubt that they come from M. tuberculosis. And the second sample, we had 11 reads from M. tuberculosis. And again, some other bacteria that we decided were contaminants and we could ignore. So in total, we got 15 reads. Yeah.
What happened to the other 0.3%? 20,000.
I'm not showing you all the data.
Oh, okay.
So actually, the biggest other component is usually either E. coli, which is in the sequencing lab because all their vectors and everything are based on E. coli, or it's a spike-in vector called Phi-X174, which is also in the sequencing lab. So that's most of the other 0.3%. So this is like 0.001% is all that we're actually getting. So we actually 1 read in a million, whatever percent that is. So we got 15 reads out of about 15 million. So we actually then can, now that we know what it is, or we think we know what it is, we can take all those reads and we can align them with another aligner. So for this we use Bowtie, which is a very fast next-gen aligner, developed, I should say, by a former student of mine named Ben Langmead. So we aligned all the reads again against a particular strain of M. tuberculosis, the closest matching one, and then we found there were actually 34 reads that we could identify, clearly MTB, so about twice as many. Still only 34 out of 15 million. But they were clearly TB, and this is not a contaminant that we see. You should not have TB in your brain. So based on that, actually, and I should say that our protocol is a prospective protocol, just doing experiments, is not guiding treatment. But in this case, the clinician who was taking care of the patient knew that we were doing this and came to my office to look over my shoulder and say, well, what's in it? What do you see? And I told him, and he changed treatment right away that day. So, and the analysis only took like about an hour. So, and I did it more carefully. It took another hour. So this is a timeline of actually what was going on with this patient. The brain biopsy was done on day 1 here. The sequencing was prepared starting around day 4. It took a couple of days because we don't have a dedicated sequencer, so this could happen in 1 day. We got the sequencing on the 7th day after the biopsy was taken, which I think was a Friday. And Mycobacterium anti-tuberculosis treatment was started right away, and the patient quickly got much better, and was in fact discharged just a few days later because she was improving rapidly. Went home, continued to get better, and on the right you see another MRI done from the same patient 3 weeks later, and you can see, although there are still a little some hints of lesions there, they're mostly gone. And in fact, she was followed up again about 5 months later, and she had completely recovered. So we think we got it. We're pretty confident that this was M. tuberculosis because the treatment she was given was tuberculosis specific.
Is it common, this kind of tuberculosis in the brain?
Not common, no.
It's a new discovery.
Oh, no. Tuberculosis has been seen in the brain before, but it's not the first thing you think of.
But this kind of effects usually are more acute, yeah?
Usually what?
Acute, there is a form of tuberculosis meningitis is very good.
This is not the usual cause of brain of neurological symptoms. Is that what you're asking?
No, but usually infection with tuberculosis of the brain is completely different symptomatics. Yeah, I know it's a very acute disease like meningitis, tuberculosis meningitis. It's very, but this is. Okay.
So I don't know enough about the phenotype of the patient to know.
Before, without the techniques, it would have been impossible to make such conclusion ever before.
Well, no, well, with TB, what the pathology lab does is they grow TB. They need to know to try to grow TB in the lab. TB is known to be a very slow-growing organism, so they probably would have eventually been able to say it's TB if they tried hard to grow it. They did a test for TB. They didn't get it, but it's hard to grow and it's slow. So they had not yet diagnosed it, but you could eventually get it. It's slow, and you can't change biology. It just takes a long time to grow.
Did they do a PPT at her first admission?
Did they do a what?
Skin test to see if she had.
Oh, this patient had TB infections earlier in her life. They knew that. In fact, the clinician suspected it might be TB, but the TB test from pathology had come back negative. And they had this nocardia test that came back positive. So they thought they knew what it was. So they were fooled by the conventional pathology. So let me tell you about a second case. This was actually an easier case to diagnose. This is PT5, a 52-year-old man who came in with lower extremity weakness, right hemiparesis, and he had a partial motor seizure. This is a brain MRI here, and you can see there's a big flare, a big white patch over on the right. All right, so I don't know much about neurology or brain MRIs, but that's not supposed to be there. That's bad. So there's something in that region of the brain going on that's not supposed to be there. And they had done PCR tests for viruses and bacteria. They're all negative. I don't know exactly which viruses and bacteria they tested for. And by the way, for those who don't know, these tests, when you're doing PCR, you're testing for a specific organism. So you have to guess what it is and then do that PCR test. And so those were all negative. They were not doing it from brain matter. I think they were probably doing it from some CSF or from blood. So we did sequencing on this patient as well. And we got more the typical number of reads. We got about 26 million reads, and you can see, I didn't put percentages here, but there were 25.9 million reads that were from human again, and we had many bacteria present. This is actually more typical. The other sample I was showing was actually cleaner. So you can see that we had 548 reads from E. coli. As I said a few minutes ago, there's E. coli in every sample. We had a number of other species that are either biological contaminants or possibly computational ones. And this is not uncommon that I will see dozens or even hundreds of different species show up with a few reads or a few dozen reads. In this case, a couple species with a couple hundred reads. But the thing that really stood out was that there were 8,944 reads from something called JC polyomavirus, which I had not seen before. I didn't know what it was at the time. I know a lot of pathogens, but there's thousands of them to know. So JC polyomavirus turns out to be a very common virus. It colonizes, we think, like on the order of half the world's population is colonized by it, but very rarely causes infections except in people who are immune compromised. This patient was not immune compromised. So immune compromised usually is because you have HIV or because you're undergoing chemotherapy, which is suppressing your immune system. Neither of those was the case for this patient, so they didn't think that there was a reason for him to have JC polyomavirus. But once we saw this result from sequencing, it was pretty overwhelming. This was clearly the dominant organism in the sample. And they did follow-up pathology testing looking specifically for signs of JC polyomavirus and confirmed that it was correct. So we got this one right as well, but we got it before the pathology did. In fact, we were able to give guidance to pathology. So the diagnosis was something called progressive multifocal leukoencephalopathy. Hopefully I said that right, caused by JC polyomavirus. There's actually no treatment for that. You just hope the patient gets better. But at least they knew what he had. We knew what he had. So those are just two cases. Our first 10 cases are described in that paper. We've done about 23 cases now.
What happened to that guy? Did he recover?
I don't want to say this wrong. I don't think he recovered. I think he died. I don't think he survived. There's no treatment for that. I knew it a year ago, the answer to that, but the patient was in the hospital a year and a half ago. I don't think he survived. A number of the patients that came through, so these are all 10 of these cases that we looked at were all really critically ill people. Many of them were actually referred to Hopkins after they had failed treatment somewhere else, and their doctors had sort of given up trying to figure out what was wrong with them. And that's how they ended up at Hopkins. So they're not local to Baltimore or anything. In fact, that patient came from another country. I shouldn't say where, but from another country. So of the first 10, yes, in the back.
What paper is this?
The paper is in the journal Neurology, Neuroinflammation, and Neuroimmunology, or Neurology, Neuroimmunology. I had it like 10 slides ago, but I can give you a reference if you want to come afterwards. And it just appeared a couple months ago. So of the first 10 cases, I showed you 2 that we solved. We solved only 3 of the 10, but I think 3 out of 10 is pretty good. There was a 4th case where some of the neurologists think we solved it. We found a bacterium that we thought was possibly causal of the infection, and they treated with broad spectrum antibiotics, and the patient got better, but we never had any independent pathology confirming it. So we just said, maybe we solved this and we don't know. There was a 5th case that's kind of interesting out of these 10. And so there were 2 negative cases where, by the way, we didn't find anything, but they turned out to be tumors. So those we got right, although I wouldn't say we solved them. We didn't find anything. There was nothing to find. The 5th case was interesting because it took about a year to get this paper published because we tried one journal, which didn't take it after dragging us out for 4 or 5 months. You all, if you're all publishing, you know how that goes. So the 5th case was the 7th patient. Turns out had a bacterium called Elisabethingia, which is not a very common infection at all. There was just an outbreak reported in a nursing home in, I think, Wisconsin about a year ago. And it turns out it looks like it's an emerging pathogen that's starting to cause more illness in the US. And because of that, the genome has just been sequenced. So when we first saw the patient, we didn't have a genome. But now there are actually 3 genomes of Elisabethingia. So when we were submitting it for the 2nd time, I went back and reanalyzed all the cases just to see if anything new came out. And this one patient, about 400 or 500 reads suddenly were appearing from this organism that had not been there before. So we're pretty sure the patient was long gone, by the way. Not in the hospital anymore, but we're pretty sure that this patient had Elisabethingia. But because the patient was long gone, we couldn't go back and confirm. But I think it's a pretty obvious case that that's what that patient had. So now for my last, say, 5 minutes, I'm going to talk about a completely different application. Oh, well, medically a different application, but the same idea. And that's to look at corneal infections. And by the way, I should say the reason I'm looking at these particular infections is because I have collaborators who have samples. I would like to use this for all sorts of different, basically anywhere you get an infection, I'd like to try this. So we've been looking for this year at corneal infections. We've done 20 cases. So here's a picture for those who don't know the physiology. The cornea, of course, right in the front of your eye. It's not like the brain. It's not sheltered from the world. We don't think it's a sterile environment. People get lots of infections in their corneas. So let me tell you about just one case, and I'll give you a quick summary of what we did. So this is a case of a woman came in with a 67-year-old woman had glaucoma. She underwent cataract surgery, which was totally routine, went fine. Sometimes you get infections post-cataract surgery, so that seems to be what happened in this patient. She came in urgently 12 days later, which was about a little more than a year ago, in July of 2015, with pain and sensitivity to light and her vision getting worse, and swelling in her cornea. Here's a picture from that time, from July of 2015. So this is not the way your eye is supposed to look. It's all cloudy. There's stuff going on in the cornea there. I have other pictures that are too unpleasant to look at, I think, so I'm not going to show you. My colleague, my ophthalmology collaborator, happily showed me all these pictures. They like to do that at ophthalmology conferences. My ophthalmologist colleague, suspected acanthamoeba infections, a type of amoeba. And they did a culture for that. So these are, by the way, the cornea cases we've done are not patients who are actually at the clinic or in the hospital. They're bank samples. We have hundreds of them. So we did 20 samples that we already had stored away. So these are no longer patients. And they're all from people who had corneal transplants. So we had plenty of tissue to use to extract DNA. So that's where we got the DNA from. For all but one of them was a corneal transplant. So this person came back culture positive for acanthamoeba. A couple days later, they gave her ibuprofen for pain, but her vision kept getting worse. They gave her drops to treat the acanthamoeba. There is a treatment for it. She was having more and more trouble applying the drops because of the pain, and the swelling was getting worse. But she continued on drops, continued to get worse. There's a problem with acanthamoeba, as I'm going to show you a picture of in a couple slides, that the drops sometimes work, but sometimes don't. If you've never heard of acanthamoeba, here's a review paper from this year about acanthamoeba treatment. So it's probably not a journal that most of you read. I don't read it. But it's ubiquitous. It's all over the place in fresh water supplies. It sounds very exotic, but it's here in the U.S. all over the place. And in fact, it was first described as a pathogen of the eye a long time ago in the 1950s, but it's most common as an infection in contact lens wearers. People who don't take proper care of the contact lenses, which includes lots of people in this country, can get acanthamoeba infections, and they can be very bad, and you can end up needing a corneal transplant if it's bad enough. And the prevalence has actually been increasing in recent years. So this patient, turns out the drops wouldn't work, and this is why they won't work. So she had a corneal transplant, and this is a cross-section of the cornea that they removed. And what you see here, all the little blue circular things, those are all little nodular regions where the acanthamoeba has penetrated into. This is a cross-section. The acanthamoeba penetrates into the cornea. So the drops basically get it on the surface, but if it goes on for a little while, it digs into your cornea and is no longer accessible to the topical treatment. So they actually they can use a corneal transplant as a way to debulk the infection, just to get rid of most of it. Unfortunately, that doesn't always get rid of all of it, because some of the acanthamoeba will migrate outside the cornea. They prefer the cornea, but they apparently live in the perimeter as well. And this patient, they did a corneal transplant, and she was initially much better, but then the acanthamoeba was still in her eye and the drops had not controlled it, so they had to do a second corneal transplant, which fortunately worked. So this is a picture of the cornea that they had transplanted in after they took it out, and it now has all these little blue dots are acanthamoeba species all through the cornea. So here's just a close-up of that. This was, and her vision was down to almost none in that eye. So she could see motion. If a hand waved in front of her face, that's all she could see. And it was very painful, and the pictures look horrible. So I won't show you any of those. But fortunately, the second corneal transplant worked. They finally got it under control. So the way that they do acanthamoeba diagnosis today is either they look for the organism in a microscope from a little scraping they'll do on your cornea, or they sometimes can take a little punch, which is of course less comfortable. They can try to culture it on plates of E. coli, but not that many people know how to do that technique because it eats E. coli, so you look for trails of the amoeba having made its way through the E. coli. Or you can do PCR molecular testing specifically for acanthamoeba. And here's a review that was published fairly recently of 121 cases of acanthamoeba keratitis. And it showed that in these cases, their average age was 41 years, that all sorts of different methods were used to diagnose it. Histology, PCR, microscopy, and then cultivation were all used in different percentages of the cases. But in only 23% of the cases was acanthamoeba the correct initial diagnosis. So 77% of the time, it wasn't diagnosed correctly at first. What's that? They were, well, I don't know exactly. I think they were definitely missing the acanthamoeba. I don't know what they thought it was, but they were missing the acanthamoeba. So they had, the initial diagnosis was something else in all but 23% of the cases. And it took an average of 3 months to diagnose. So as I just showed you, if you take, as you're waiting to diagnose this, the acanthamoeba are crawling deeper and deeper into the cornea. You don't want that. So we did sequencing on this case, and we actually did 20 cases in all. I'm just showing you this one. We actually multiplexed them all on 2 runs of an Illumina NextSeq, so it cost about $750 per sample. Took about a day to do all of them. Here, because we're using a NextSeq, which is a slightly newer machine, we got about 40 to 50 million reads per sample, which is really more than we need, but that's fine. So for this particular case, we got 46 million reads, of which 46 million were human, 99.9% or something were human, and a whole bunch of other bacteria and random things. You see that the top hit was something called Cosuria rhizophila, which is another common contaminant. It's in reagents and it's in labs a lot. So there were a lot of reads from it, but we saw it in all the samples. So we realized there's nothing interesting about it. But we saw 27, almost 28,000 reads from acanthamoeba, which was very unusual, and we did not expect to see that. So we were very confident that this was acanthamoeba. Oh, I should say that the way we did this experiment was the ophthalmology and pathology collaborators prepared the DNA, sent it for sequencing, didn't tell me anything about what was in any of the samples, and then said, now I just got the data directly from the sequencing lab, and then I told them what I thought was in each sample. Me and my postdoc, one of my postdocs. So this one was a pretty easy case once we looked at the reads. Here's a summary of all the cases. It turns out that corneas, although they're not a sterile environment like the brain, at least the cases that we were testing on here were easier than the brain. We did much better. We got 17 out of 20 with no false positives. So there were 4 controls. I'm counting them in the correct ones. There were 4 controls that didn't have an infection, and we didn't see anything in those. There were 5 fungi. We got 4 of those. The one that we didn't get turns out to be there's a fungus out there that infects people, infects eyes, that we don't have a genome for. So there's no way we can get that, although I'm now thinking, like, how do I get that genome sequenced? There were 3 acanthamoeba cases. We got all 3 of those. And a couple of bacteria, non-mycobacteria, a couple of mycobacteria, a weird thing called Ancalea algeriae, which is a microsporidium, which was very obvious in the sample. The original case, when it had come through, they had to send it to the CDC to figure out what it was because it was so unusual. And we did miss, there were 3 herpes simplex infections, and we missed 2 of those. We're not sure if that's because there wasn't any herpes in the sample anymore or not. So one last thing for my last couple minutes, returning to the computational challenges, which is what we can do the most about in my lab. So just to look at this acanthamoeba again. So here's a couple of pictures of acanthamoeba. That's what they look like. They're little creepy crawly things that you don't want in your eyes. So there are 11 acanthamoeba genomes that have been sequenced. They're all draft genomes. And I should say that every eukaryote, with I think one exception of some small single-celled organism, every eukaryotic genome that is available today is a draft genome. That includes the human genome. You've probably heard many times over the human genome is finished. You've probably heard announcements about other genomes. They're all draft genomes, meaning they do high throughput sequencing. I'm involved in a lot of these projects. We assemble the genome. It's in a lot of pieces, but it's good enough, and we publish it. And we say we're done, and then all the glory is gone, right? I mean, the job of finishing it and filling in all the gaps, that's kind of painstaking, and the paper's already published, so they often sit that way indefinitely. The problem with draft genomes is that they're noisy. They have bad data in them. So when we first ran our first 10 samples from the corneas through, this is showing you along the x-axis are the 11 different acanthamoeba species we had in our database. And all 10 samples had some reads from acanthamoeba. The scale, you probably can't read, this is 10,000 reads, 20,000 reads, up to about 50,000 reads. So they all had acanthamoeba, and we thought well, they don't. We know they're not going to all have acanthamoeba. But this is before our colleagues told us anything. So that's just a false positive. But that's very frustrating. So what we did was we took the reads that were matching. So we took the same. We took the reads that were matching acanthamoeba from another set of samples, actually from the neurology samples. We didn't think any of them had acanthamoeba. So we ran those through this database, which we've now added acanthamoeba to. And we took all the reads that matched acanthamoeba, and we figured out where on the genome does it match. For each read, where is it hitting? Because if there's some contamination, then probably the reads are all hitting that little bit of contaminant, those little bits of contaminants. And we can just get rid of those. We can clean up the genomes in our database and then rebuild our database. So this is now looking at those same 11 genomes. The red parts of each bar show you how much of the genome was hit by some reads or other. And in most cases, it's just a tiny little amount. So we just deleted the red parts from the genomes, just got rid of it and said, OK, now we hope we have a clean version of the acanthamoeba genomes. Let's go back and look at the corneal samples again. And we did, and we got this. So we decided these 3 samples really did have acanthamoeba in them. And those, in fact, were the 3 true positives. So there were another 10 samples which didn't have acanthamoeba, by the way, and we didn't see it in any of the other 10. So we only saw it, after this cleaning step, we only saw it in 3 samples, and it was the right 3.
Contaminated by what? What kind of contamination?
So this procedure doesn't tell us what the contamination is. So it could be low complexity sequences that we were unable to filter out. It's probably that. It could be E. coli, because people doing draft genomes have bits of E. coli in their genomes. It could be sequencing vectors, and it could be human. So there's a lot of different things that could creep into a draft genome. But this procedure, by basically treating hits as if we know they're false positives, just get rid of that part of the genome, we cleaned it up. So we can go back and try to figure out exactly what those were. So let me just conclude. So where do I think this is going? So this is, we're doing prospective studies now, and I'm doing what I can to try to convince colleagues around the medical community that we should be doing sequencing to diagnose infections. We should not be content to go to the doctor and be told you have some kind of infection, go home and it'll get better, or here's a broad spectrum antibiotic. We should certainly not be content when somebody that you care about is in the hospital. It's happened to me, it's probably happened to some of you, and they have some hospital-acquired infection and the doctors don't know exactly what it is. And they have some guess because something's going around and they'll guess it's that. That's the way we do diagnosis today. We sort of guess and then maybe test, or guess and then treat. That's a terrible thing to do, but we've been doing it for decades, and I think sequencing should change that. So sequencing, I think in a few years, it's certainly going to be easier then than it is today, and I think doing a sequencing assay should be easier or as easy as doing a blood test today. We will take some sample of the tissue, unless it's in the brain, of course, then you have to do a biopsy, but if you can get that tissue, we should sequence it, and we should compare it to all the pathogens and figure out what the patients have. And except for novel pathogens, which of course we can't identify by looking them up because they're novel, we should be able to figure out precisely what infections we have and what infections our patients have, and stop this what I consider to be a mid-20th century process of guessing the pathogen and treat based on the guess. So just I want to just conclude with acknowledging some of my collaborators. The computational work here is mostly the work of Florian Breitfieser, a postdoc in my group, Jennifer Liu, a grad student, and Derek Wood, who wrote the Kraken program, is now at a company. The neurological work was led by the sample collection. A lot of the analysis was led by Carlos Pardo, who is a neurologist who I work with very closely, and a number of other clinical colleagues. And the corneal infections are a collaboration with Charles Eberhard. And I'll stop there and take more questions if you have them. Thanks.